Good. So we are about to start. Everyone, welcome to the second day of biological data science. I hope everyone agrees with me that yesterday we had a really good day with a really diverse and interesting set of talks. I think the bar has been set very high, but I'm looking forward to today. And it was also great to see that many people interacted in Slack and in the social breakouts, even though we are not meeting in person. And just a reminder, we have lots of posters available in Slack. Please do go and visit them during the day. So to start day 2, today we have our first keynote session. And co-organizing this conference has been really a great experience, but being able to introduce our next speaker, who is someone who I really truly admire, is the cherry on the top for me in this conference. Our first keynote speaker is Professor Sylvia Richardson. Barbara, Mike, and I are really grateful that she accepted our invitation. We know she's very, very busy, especially these days. Now I have the really hard task of introducing Sylvia's achievements in just a few minutes. Sylvia has a PhD in probability theory from the University of Nottingham. She has held academic positions at very prestigious institutions such as the University of Warwick, the French National Institute for Medical Research in CERN, and Imperial College London. In 2012, Sylvia became the director of the Medical Research Council Biostatistics Unit and the chair of Biostatistics at the University of Cambridge. Currently, Sylvia is also the president-elect of the Royal Statistical Society and co-chairs the newly created Royal Statistical Society COVID-19 Task Force in partnership with the Alan Turing Institute and the UK's Joint Biosecurity Centre. And I'm sure that's keeping her very, very busy these days. Throughout her career, Sylvia has received many awards. Among others, this includes the Guy Medal in Silver from the Royal Statistical Society and a Doctor Honoris Causa from the University of Oslo. She's also a fellow of the Institute of Mathematical Sciences, the International Society for Bayesian Analysis, and the Academy for Medical Sciences. In 2019, during the Queen's Birthdays Honours List, she was also appointed as the Commander of the Order of the British Empire for her contributions to medical statistics. And she received her medal from Prince William. And as part of her research, Sylvia has made many, many significant contributions to Bayesian statistics with applications to the health sciences, focusing on complex and highly structured biological and epidemiological data sets. Among others, her recent research has focused on developing methods and scalable algorithms for clustering and high-dimensional regression, as well as application in single-cell transcriptomics and EQTL analysis. To date, Sylvia has supervised more than 40 early career researchers, including PhD students and postdocs. I'm very likely to be part of that list, and I can certify that beyond being an excellent researcher, Sylvia is also a great mentor and is always keen to support the career development of all those who work with her. Personally, I'm very grateful for the support and encouragement that she has given me during the last few years. I can honestly say that I would probably not be where I am if it wasn't for that. So, well, I think it's time for me to stop talking and let you enjoy Sylvia's talk. So without further ado, please join me in welcoming Professor Sylvia Richardson. And Sylvia, please go ahead whenever you are ready. And just one minor thing, thanks for the clap. Sylvia has agreed that if you want to tweet or take photos of her talk, that's all fine. So, Sylvia.
Thank you so much, Catalina, for this very nice introduction. And I must say that it was so enjoyable working with you and seeing you, grow into such a fantastic researcher that you are now. And I also wanted to thank Michael and Barbara for inviting me. I've been to the Cold Spring Harbor meeting once before in situ and really enjoyed it. So here what I want to talk is about two approaches that we have recently developed on hierarchical models for but the background to this research is that, for a number of years with Leonardo Bottolo, spurred on by the increased advances in biotechnology, we have tried to focus on joint analysis of association analysis of multiple multiple phenotypes, and on trying to do that to improve the power basically of the analysis by actually trying to do this joint modeling. And so basically, you know, the theme and some aspect of the modeling approach what I'll present today stem really back to this long-standing collaboration with Leonardo Bottolo. And then Hélène Ruffieux joined the project while she was doing her PhD in Lausanne with Anthony Davidson. And she's now based in Cambridge as fellow, research fellow. And she really, I just want to really acknowledge her huge contribution to the project, and particularly forward a scalable interpretation and some really nice biological example. So basically, you know, we all know that genome-wide analysis.
Sylvia?
Yes?
Sorry for interrupting, but the sound is a bit fuzzy sometimes. So maybe if you switch off your camera,
Shall I try to?
Just turn off your video camera.
Okay, I've done that now. Is that better?
Yeah.
Okay.
So far it's good.
So far, so good. Yeah.
Yes. So basically, so what I was saying, genome-wise association studies to study we're very familiar with that, and there's now been an immense number of associations found.
Yeah.
Is it still bad?
You were breaking up again. Are you using Wi-Fi or are you using?
Yes, I'm using. I can't do anything else.
All righty. Thank you.
I'll try to speak closer to the.
What kind of microphone is she using?
I'm just using my computer microphone like usual. Is that better now? Or?
Yes, go ahead. All right, we'll just have to deal with it.
And so basically, a lot of these associations which have been found are not in basically in conclusion. And so it's quite hard to actually try to decipher the mechanism by which the association could occur. And so a lot of research has now been developed in utilizing intermediate levels like transcriptome to try to decipher the mechanisms. So the type of questions, as I was saying, that we try to address is to find genetic variants which control multiple traits. This has required tailored modeling approaches, and we've used the powerful framework of Bayesian hierarchical model to do that. Now, we have a of covariates, a large number of SNPs, but we also have a large number of traits, so of responses. So we're facing basically two large-dimensional. Is quite low because these are excessive and so they're not correctly done. And so we have to manage the statistical tension between which is our statistical aim, and nevertheless computational concerns to make the tools implementable and useful. So here I will focus on the and by that I initiates decisive functional mechanism. And here I two recent review papers which discuss how using functional genomics will help to identify a mechanism of complex disease. So we have two challenges. One is to detect these EQTL or PQTL or MQTL genome-wide, and another challenge is how we find the interpretation. So for doing this detection, genome-wise, we have developed ATLASQTL, which takes as input the usual two matrices of the SNPs on samples, and the molecular traits which have dimension Q's, the POs in the same dimension and Q the traits. And what it returns is posterior probability of selection of this hotspot, highlighting all the traits which are linked to one hotspot. Here we've distinguished the cis one and the trans one, but basically we're focusing on trans hotspot. And this information is summarized below here with a sort of Manhattan plot where you see the hotspot location and the number of traits associated with them. Then to actually dig deeper and try to understand mechanism, we have developed another sort of model building an ATLASQTL very recently, which is called Epispot. And this time the data has now got additional external information on each of the SNPs in terms of a large set of epigenetic marks. And then the output will return, and then these will be incorporated jointly to actually help to find the hotspots, but also try to interpret them. So we have, we're selecting additionally the relevant marks for the hotspots which are analyzed. So basically, I talk about first, there's a commonality. I will try to highlight just key aspects of the modeling and key choices important in the hierarchical Bayesian, discuss a bit how we're doing this computation, and then I will illustrate that collaboration with Fairfax. We have been working on this analysis. So the first stage of the model of ATLASQTL is a model where we're linking, so we have Y, which are traits, Q, the index ET, right? And then we've got P SNPs. The index will be S, and we are doing a linear model. And on the coefficients, we have putting a usual spike and slab model so that we can select which SNP is associated with which traits. Underlying this, there's probability of association, gammas, this indicator of association for the actual slab part, which are with omega st being the probability, and that's what we are focusing our attention. So that's a usual first stage model. We are allowing for some variability specific variability specific trait parameter. And here this model is represented in a usual DAG form where you see basically the regression parameter, the selection indicator, the underlying probability. Now, how do we model the whole thing, information across the traits, i.e. across the actual T? We need to think about the model for the omega. Now, we have introduced here a second level model, which uses a probability model for basically these probability. We have both a specific effect and a trait-specific effect.
Sylvia, I'm really sorry, but it's breaking out again. I'm not sure. Is it are you using the microphone from your laptop or?
I am using my microphone from my laptop. I've never had that before. I think it must be because
Are
it's
there
the
other
same.
apps open on your machine that you can close to?
I could try, but then I have to get out of that, stop sharing. This will take a little time, I'm afraid. I could have, I don't think so, honestly. I can't see anything open. I'll close my email, but there's nothing else open.
Or Internet Explorer as well.
You know, this is your link and that's it really.
Yeah.
You want to try and share again?
Yes, I'll try to share again.
Still sharing with us?
No,
Okay, ask her to share again, please.
done.
I mean, it sounds totally fine right now. It's weird.
Yeah. We're talking for like 20 minutes, completely fine. If not, Sylvia, you can email me the slides and I can show them.
Is that, I'm sharing now, but I don't know if it's in full screen.
It's not
There.
on full screen now.
Yeah, okay, is that better now?
Yeah, now it's working.
Good.
Sorry, so I was trying to explain that we've got these two parameters, a sort of column effect, which is a parameter which is shared by all the traits linked to a SNP, so theta S, and then a trait effect. And so it's how we're putting priors on those, which is really important. I want to share some information. So if I discuss first the trait-specific parameter, this will try to adapt to the sparsity level corresponding to each response. And hence it has the normal distribution with these parameters, which are specified thinking, but it's really not that differentiated. What's more is how you share this theta S parameter, so which is sharing across all the trait some kind of propensity to be a hotspot. So this is like a hotspot propensity parameter. Now, it's the variance that you would put on theta S is influential, and one has to be very careful when there's little information. If you're just having a fixed variance, so we have we have used actually a rather flexible model.
Hey, Sylvia.
Yeah, Sylvia.
It's all garbled. It sounded a lot better when you were not. I don't know why, but it sounded better when you were not at full screen. Can you just try looking at? Yeah, drop out of full screen.
There.
That's I don't really understand why, but it seemed better. I don't know if the
okay. Maybe the full screen takes the
yeah, maybe it was just Acrobat was being weird. Let's try this.
Yeah.
Is that okay now?
It seems okay, yeah.
Yeah, it seems okay.
Oh, I'm really sorry. What a mess. Because I'm losing the thread. Anyway, shall I start again on theta S? Was it totally garbled or?
Yeah, why don't you start there?
If you start again, yeah, thank you.
So basically, the theta S, which is really the crucial parameter, this hotspot propensity, I was saying that fixing a variance for that is not really a good option because it can lead to real piling up effect when you've got a very large number of traits. And so you have to try to learn that variance. And we have found that putting a horseshoe prior on it, which means that the variance has got a global parameter, the theta 0, which we're trying to learn, as well as a local effect, which is very important to be able, and both of those having Cauchy distribution. So this local effect, most of the time will be, nothing, but it's allowing large hotspots to survive this kind of massive shrinkage is going on. But nevertheless, the whole shrinkage is controlled by this global parameter. So we have found that this using a horseshoe prior. So horseshoe prior have been used directly on regression coefficient we're not using directly on the regression coefficient, we are using on the selection indicators. So this is quite a different way of using, but because of the kind of nice property and robustness, we have found that they really work well. And just to, so now how do I go down there? So just to show, you know, the horseshoe is because the name is because the shrinkage parameters should have this horseshoe, i.e. shrink most of the time and let a few things escape. And what we have found, which I want to highlight, is that in terms of the global sparsity parameters, it's useful to not to fix it to a fixed value, but to a value which is related to the number of traits you're analyzing. And there's some theoretical justification for that. And so basically here we're having the scale parameter of the Cauchy, which is related to the number of traits. So it does kind of an implicit multiplicity adjustment. So I think I'll go quite quickly in terms of the variational base, because we have lost some time, and I do want to get to the example in Epispot. So we have basically done a variational base version with so a product, a mean field approximation, but of course you do not want, you want to preserve the mixture prior, so you do not assume independence between the beta and the gamma in this approximation. But because we're in situation, we're trying to select among many correlated SNPs and covariate, there are issues about you know, getting stuck in local modes. And so we have found it necessarily to develop an anneal, to add a simulating annealing process on top of the variational base. Which doesn't create any complication in the usual way as you do annealing. And to show you the, on a simulation study, the performance of ATLASQTL, where we have 20,000 traits, 200 SNPs, and 400 samples. So you see a massive number of traits. And here, the true hotspots are little red crosses. And you could see that we are really recovering them well. On the other hand, if we were running the version without annealing, most of the time it just gets stuck in local modes, and we're very far from recovering the truth. So this annealing is, when you're using variational base in this context, seems absolutely necessary. We've compared with our previous fully MCMC approach, HESS, which we developed with Leonardo, where it's a similar model, but we do not have this penalty, we do not have this variance adjusted penalty for the hotspot propensity parameter. And as you could see, the fully based model, it's really not scalable with respect to the variational base, 5 days instead of 2 hours. And ATLASQTL has an improved performance thanks to this, you know, this flexible modeling of the variance of the propensity. And of course, there are both approaches beat one at a time QTL analysis quite clearly. Here is an example where we've run genome-wide on monocytes EQTL. So there's 24,000 CD4 unstimulated monocyte transcripts. And we have 380,000 SNPs, and we have run in big blocks, LD blocks, basically in parallel. And here I'm showing you the whole hotspot map of the genome, and we see there's a couple of very high hotspot involving between 200 and 300 traits. Here we've used an arbitrary thresholding of a posterior probability of inclusion for any of the association larger than 0.95, but this can be calibrated by permutation. Now, there's a particular place of interest here, which actually is a known pleiotropic locus near the Liz gene on chromosome 12, which has been reported in several papers, and of course also by Ben Fairfax. To date, the functional SNP is not quite identified, neither the mechanism of action. And so I'll come back to that towards the end of the talk. At the moment, the SNPs in that region, which have been reported as current hotspot, are these 4. And there are 2 genes, the Liz gene and the Now, we've got now about 300 transcripts, which I linked to that region. Sorry, I think I'm losing signal again a bit. No, it's okay.
Sylvia, it's fine.
So we've got a set of genes associated with the hotspot, and we try to understand, are they directly associated? Are they mediated through the cis gene, for example, Liz or something? And to try to understand a bit more how this I've also looked at the conditional independent structure of this set using a Gaussian graphical model approach, which tests basically conditional independence, i.e., the dependence between 2 things, conditional on all the rest, which we have developed with Gwen Ledet. And there's a software BEAM to actually do that. And we've actually applied BEAM both on the original transcript. So after, we've done the hotspot analysis, we've got that set S, we could do the graphical model of directly this S transcript, or we could have the residual value, which are computed after the regression on the SNPs. And it's a massive network, so I'm only going to show you the subnetwork of the transcript in S, some statistics to show that it's particularly dense, as you would expect. It's got 70% in the raw, and the number of edges diminishes when we're looking at the residual, but it's still very high. As you compare, for example, the node, the histogram of the distribution with respect to the rest of the network for this particular set is very, the medium is way high. It's a very large network. So if we look around to try to understand, is the effect of the hotspot mediated through Liz or Yeats? We've looked, you could see indeed that Liz and Yeats are connected to a lot of transcript by definition, well, not by definition, but in this network. These are direct connection. There's a conditional independent graph, so these are direct connection. But interestingly, when you actually look at the residuals, so you do exactly the same analysis, but not on the raw expression, but on the residual expression, we find that more or less all the connection of Liz, Yeats has no more connection. Liz has only very. So what we've done is we've conditioned on the hotspot, and this has completely changed the network, which is really not in favor by saying that the control of that trans network goes through either Liz or Yeats. So there's no evidence of that, and we have to dig further, which we will do thanks to our Epispot model. So I'm going to come back to that example at the end. After explaining the basics, additional assumption that we make to try to actually use information from epigenetic marks. So the first level model is exactly the same as in ATLASQTL. So I don't have, so I'm just re-showing you these. So we have, again, regression of the traits, and then we are interested in this association probability between a SNP and a trait. But it's in the modeling of these association probabilities these indicators there, that there's a difference because we're now introducing additionally to the column effect for the hotspot propensity, to the trait effect for looking at the variable, at the trait specific characteristic. We also have now a regression on a set, which could be quite a large set of marks, epigenetic marks. And because we here do not want to pre-specify a small set of marks to include in the analysis, but we want some kind of model-free again, we are also implementing variable selection at that level. So again, a spike and slab formulation. So there's a spike and slab formulation at the level below, and now there's within here a spike and slab formulation of which marks are going to be relevant for this set of transcripts and SNP basically. So this Xi models the involvement of a sparse subset of epigenetic notation, because and you could see that we've now got 3 variants to estimate. This new variants coming out from the epigenetic marks, basically selection, and then the propensity, the hotspot propensity, and the trait-specific variants. And for doing that, and for computation, for ease of computation, and because now we're at this, we're selecting at the second level, so we're even further up in the hierarchy and how information is kind of shared between all that. We have used empirical base using a new version of variational EM algorithm to actually estimate these, and we haven't gone to add an extra level. And we have found that this works well, because what's important here is to have this very local effect, so we have kept here a local index by S variance for the hotspot propensity. So this looks really complicated, but basically, if I just try to recap what Epispot does, it does a hypothesis-free selection of a large panel of annotations. So you could introduce 500 annotations, and it will just pick up a few of them. And these annotations are informed and helping to do the QTL mapping. So they give you more power, but if they're not relevant, you don't lose. We have done simulation, and you don't lose much because we have this sparse selection. We're not imposing them. We're actually having this selection, which means that you could more or less discard them if they're not relevant. So our tool is really tailored for interpretation of the hotspots. But what I want to actually stress is that annotation needs to be sufficiently represented at causal variance. So this is not for doing genome-wide. This is when you have actually run a selection of genomic region where you think that, where you know that there are some loci of interest which you would like to understand more and refine, and then you run Epispot. This is to the difference of ATLASQTL, it is not a genome-wide tool because you need quite a lot of information. The marks need to be informative to be able to find something. An interesting aspect, which I don't have time to detail, is we can actually look specifically at at modules. So we have a version of the model which is module specific for detecting the mark. So if we want to break down a set of traits into modules, then we can have mark specific to modules. So now let's go back to, well, first a simulation study to show you that, and there's many more in the papers, to show you that it actually does improve the analysis. So here we had 600 simulated traits and 500 epigenetic annotation. And we assume that we had 60 loci each comprising of either 0 to 3 active SNPs, and that the active SNP were associated with typically 27 traits or something. And among these 500 annotation, only 3 are simulated to trigger the QTL effect. And we picked up the 3 quite easily. And you could see that in this case, because there is information here, the ROC curve is, of Epispot is better than the ROC curve of ATLASQTL, both for detecting the association, the pairs association, and also the hotspots. And now to end, I just want to look at, go back to our monocyte analysis. So as I said, Epispot should be used after there's been some pre-screening of interesting loci. So we've used the Fairfax data set, the one I had presented in the first part, to do the screening. And then we've used another data set, the CDA data set, to actually refine and try to understand, in particular around Liz, what was the, what marks and what could be the mediating genes. So after mapping between the Fairfax and the CEDAR data sets, we end up with a pre-selection which includes 474 traits, 195 candidate loci and windows around it. So we are running with 1,500 SNPs. And here the sample size is a bit smaller than Fairfax, which was 400. It's around the 300. And we've considered a large number of annotation, including DNA sensitivity sites for managed tissue and cell type, ensemble gene notation, chromatid state data from ENCODE, and so on. And we find here, again, the hotspot. So this is a different data set than the Fairfax. So we again find on chromosome a hotspot around the Liz region, which I'm going to analyze in more detail. And interestingly, there's among the DNA's annotation, it's the monocyte specific ones which have come up as being selected. So strengthening basically our hotspots there. Now, so this is basically, there's 4, there's a few candidate SNPs in that region which have been in the literature. And Epispot selects a monocyte specific annotation, DNA's annotation. And there are 2 candidate master SNPs here. And interestingly, if we look at the network controlled by these 2, which are probably around a functional SNP, they're together associated, there must be tagging a functional SNP, we find that the gene network controlled by this hotspot is enriched in CREB1 transcription factor binding site. So this is, I'm really describing very quickly a lot of discussion that has taken place with all our collaborators. And we also find that these hotspots are strongly associated in trends with CREB1, who is on chromosome 2. And so again, the idea is now, because it doesn't go in, this trans network is not involved through Liz, but could it be, could CREB1 be a distant mediator of this hotspot transgene network? So again, we do our network analysis. We see that CREB1 is linked, it has a lot of links on the raw expression. And this time, interestingly, when you account for the effect of the hotspot, if you remember beforehand, more or less all the links had disappeared. Here, basically it makes no difference. So not giving no contradiction to this hypothesis that the hotspot may be controlling this whole large group of transgenes through CREB1. Of course, to really validate that, you need some experimental thing, to run some experiment, and we're discussing that at the moment with Ben Fairfax, whether this is actually possible to do. But we are, thanks to the fact that we use these marks in Epispot, we have found an interesting hypothesis around this trans network, and to explore further. So basically, I'm at the end. So just recapping that we have two complementary hierarchical framework. One, the second one building on the first one. One is really for genome-wide detection. The other one is for refining around a set of loci. But even though I've illustrated that on transcriptomics and expression QTL, these approaches are generic and can be applied for discovering QTL into protein or to metabolites. So in terms of the distinctive statistical features, we have used joint Bayesian hierarchical model, and borrowing information has been key to increase power to try to focus on these trans. But on the other hand, when you'd always faced with a lot of variance parameter in these Bayesian hierarchical models, and great care has to be taken to actually investigate how to actually estimate or do a hyperparameter modeling of these variance parameters. And that has been very fruitfully done in ATLASQTL, where we've got a careful discussion of how to do that. And then on the scalability issue, we have found that variational inference is delivering in this context, but when you're doing variable selection at such a large scale with complicated correlated features, it's necessarily to think about annealing and simulating only on top, otherwise you get stuck. So here I will stop by giving you reference to the two papers. Epispot is already on bioRxiv, and the actual code are available from Elaine's GitHub page. So I want to acknowledge again Elaine and Leonardo, my close collaborator on the statistical side, and then Ben Fairfax and Isa in Oxford, as well as Elena Vigorito and Chris Wallace in Cambridge, with whom we have a very fruitful discussion about interpretation and non-interpretation of the hotspots. So thank you very much. And maybe I should stop sharing now.
Yeah, thank you so much, Sylvia. That was a wonderful
I'm so
talk.
sorry that, let me put my video on. I'm really sorry that the audio was bad.
Don't worry. Once you stop having the full screen, everything worked okay. So
Okay, that's
don't
good.
worry.
So luckily,
We could hear you.
luckily we
Good
actually, yes.
Yeah. So we are waiting for a few questions from the Q&A, but in the meantime, I have one. It was really great to see. I remember the early stages of some of this work a long time ago. So it was really great to see all the new advancements. So I know in your talk, you focus on EQTL analysis where you have your responses continuous, but there is a lot of people trying to look at things like in UK Biobank, or there are different types of outcomes that are non-continuous, like binary or time to event. Do you think some of these ideas could be applied or translated in that context?
Yes, you mean if we just have a sort of mixed class of molecule? I mean, I think the difference is, are you trying to focus on a few, for example, categorical, something close to the disease description? Or are you trying to incorporate a high level of molecular traits, which are typically, of the same kind? I think that there's quite a difference in terms of the modeling approach. If you are focusing on, which is what we've done so far, if you're focusing on a few combination of clinical traits, which could be like blood pressure, as well as some categorical variable, then you need to really develop latent model of some kind of, and this is doable, but you need to kind of develop a latent model here to actually of latent quantities which are linked to those, and then you could apply that setup. But I would say, and I think Leonardo Bottolo has definitely done something in that line recently. So I think there is a paper which has been accepted. I don't have the reference there, but if people are interested. But it's quite a different setup in a way, because the hierarchical nature of borrowing all this information becomes much less strong in a way. If you only have a few clinical things, then you jointly model them together, but you don't have this context where we could only do that by having this hierarchy in a way.
Yeah, no, that's true. Yeah, I was thinking like a few kind of clinical phenotypes,
So
like UK Biobank related.
You could certainly do, but in that case, it's less the sort of all these random effect sharing propensity thing become less relevant.
Yeah, thanks, Sylvia. So we have a question from someone from the audience. So Aaron McKay is asking, can the Epispot model be expanded to include multiple layers of molecular hypotheses, as in your introduction slide?
Yes, I mean, the Epispot and the Atlas could certainly consider different groups of traits. Is that, if you're asking whether the Y could be different layers? Yes, definitely. I mean, and in particularly, Epispot has got this module specific option. So you could have, which means you could group blocks of traits and look at the epigenetic and so on. You're running in a way separately on these different blocks. So you could have proteins, and you could have a block of transcript and a block of protein. I mean, there is already in ATLASQTL, this is possible because we've got trait-specific variances, for example, and parameters. So, to account that some could have very different level of variability. So you could pile them up, and run the lot. You'd have to be careful about looking at whether these trait-specific variances, it wouldn't be very difficult to actually sort of block them in a way to account for difference. I think that would be pretty not that difficult. And in Epispot, as I say, we have these modules already built in. So the modules could be modules of different type of data. It could be protein and transcript.
Thanks, Sylvia. And we have a follow-up question from Aaron. So he's asking, pleiotropic genetic effects do allow for compounding signals to better power discovery of master regulations. How will you approach monotropic genetic effects where there is pleiotropy occurs downstream?
I think that's beyond, I don't think I completely understand. You mean the...
Aaron, do you want to?
I don't quite...
Aaron, if you raise your hands, we can promote you to panelist, and maybe you can ask your question.
I don't. So you wanted to a pleiotropic effect which occurs later downstream, you mean? Is that?
So he's asking, single SNP controls single downstream gene. The gene then is a master regulator of many others.
Yes. Well, that's what we are modeling.
Yeah, so you're sharing this information by having this hotspot.
Yes. So basically,
Aaron, do you want?
the master, we are assuming that we're looking for whatever point in the biological process, we are looking, our framework. So it doesn't have to be SNPs. Yeah, if that's your question is, it doesn't have to start with SNPs. What we have is a set of predictors, a large set of predictors, a large set of which can start by being continuous predictors. They don't have to be SNPs, they could be any predictors. And what we're looking is downstream of that, whether some of these predictors influence a large number of the downstream features. So it can start at any level. Indeed, so you could look at something between transcriptomic and proteomic, if you wanted to do so. The traits would be the proteomic and the X would be the transcriptome. Does that answer your question?
Yeah, Aaron says yes. Great, thank you.
Kata, can I jump in here? So I had a
Yes.
question. So in the Epispot, if I understand correctly, there's an EM algorithm for some of the parameters, but then there's you also use simulated annealing to kind of further refine them. So could you comment on what the virtue of the simulated annealing is?
Well, I mean, we've done the EM algorithm because we wanted to be able to practically to also run in parallel, and we have found, I mean, there's other ways you could actually run that inference. You could go back, but because we are at a level, we are doing variable selection at the next level up. So among the marks, we wanted to have some kind, like we could imagine doing a horseshoe at that level,
Right.
If that's what because we've had a horseshoe but on now the selection of the marks at the second level, we could imagine, doing again a horseshoe at that level if we wanted to be totally, coherent and sort of, but we found that, this is pretty clunky. This could be, we didn't even go that way because we wanted to be efficient and not, we are very high in the hierarchy and information is scarce. So that's where we find that using empirical based approach, which usually gives a good approximation for these variances and keeping the bit of the variance, like the local nature of the variance for the hotspot, which we've learned in a way from ATLASQTL, was actually performing pretty well. And so that's why we went for this EM, so not to have an additional layer to actually, so to run,
I see.
to run priors. So we went for basically empirical base, and which implemented through this EM variation. The code, it's a pity. Yes, well, it's detailed in the paper, and I could send you. I had a couple of slides on that, but I don't think it's hard to read.
Okay, I'll check out the preprint on that.
Yeah.
Yes,
Sylvia,
but
if you want me
basically we decided, we could have actually gone again for a horseshoe, that would be one choice. But I trust Hélène, who said that she thought this would reach out, this would really stretch a little bit the whole exercise, and that, we also didn't want to fix something for the Epispot part, we wanted to be coherent a whole model. So this is why at that point we decided to do all these three variance parameter with an empirical based approach. And and we could go back in a way, and we haven't done that, and compare where this would be enough in ATLASQTL, for example. That's something maybe we could do.
And then I guess as a follow-up, where do you see the future really lying? Is it just larger numbers of samples, integration of multiple data types, I don't know, better modeling?
Well, I think I think it's really, in terms of, for me, it's a strategy. I think you need to have good tools to answer some questions. So I think we've got the tools now for this trans, but it's the strategy of how to use that successively, combined with network analysis, to actually have a sort of strategy of analysis and try to define that in a way a priori and then try it on big case studies. I mean, that's what I think is the future, is trying to think globally and good strategy about, you start with that, you get the thing, you actually integrate that data, then you go forward, and not in a, because I think, and maybe explore different types of strategy if you have a rich enough data set where you have three layers, for example. So you'd have also proteomic. And then, you have many choices, you put them together, you start getting the hotspot to there, and then you integrate the next data set and you compare and so on. So I think I would love to have a sort of big, meaty biological question where we would have this joint model and then we could compare different strategy, because the way the literature goes, everybody follows their strategy, but they haven't thought about what's the strategy. It feels sometimes, like all of us, we just go one way, we just go in one route. And I think it'd be really good to...
What do you think integration of like having widespread proteomics and other sort of layers of data, so you can kind of
yes, and I feel that the networks have a big part to play to try to home in on interesting parts, because I think trying to analyze everything jointly, you just lose completely the signal. And so, this network analysis that we've developed, which look at conditional independent graphs in thousands and thousands of, could cope with 20,000 nodes, no problem. At least, it can help you, and then you could extract down sub-network, and it can help you to actually also focus the integrative analysis. So in a way, I see that there's a need to possibly, if you've got full, very large integrative data sets, I think you maybe need to do a first screening of some part where there's some action, there's lots of links, there's some first, and then go in that type of analysis that we've done, which kind of try to refine where the association are and so on. I think the combination of network analysis with regression type of analysis is useful.
Yeah, I think so too. Thank you.
Thank you. Yeah, and I guess many of these techniques that you described might start be useful now in the era of the single cell EQTL analysis, where people are looking at related cell types, perhaps trying to share information, especially in that context where you have very sparse data sets. Maybe these type of approaches will be very beneficial.
And I think Leonardo and Hélène are trying to develop a sort of Poisson version at the moment as well, so for Poisson data, which would be more appropriate for single cell. And then we have to see whether, this is too clunky or there's some approximation or how, but I think that's an important area of application too, yes.
Yeah, it will be really great to see there. I think there are lots of opportunities coming up of there, and maybe variance QTLs that people are starting to see that so far people haven't been able to find many of them. Maybe the type of approaches that you are developing will increase the power and enable us to
definitely it increases the power for trans in a non-single cell. I think definitely, because we know there are weak effects. So the idea of trying to look for that propensity to link them really boosts, as you've seen on the ROC curve, it considerably boosts the power. I must say, be very, the two pieces of software are available, and we'd be very, and the paper is very interested to hear comments from people trying them and so on. So please feel free to contact us, to contact Hélène and so on questions, or, let us know your experience with trying them, because we try to keep improving them.
Yeah, Sylvia, if you send me the links, I will put them on Twitter and in our Slack channel so that everyone can find them very easily.
I'll do that straight after.
Yeah.
Okay, well, thank you so much for your time, your wonderful presentation. Yeah, we'll definitely promote the links over Slack and everything. But sadly, we